In this slide set, we will explore the structures and functions of genomes, including the genomes of eukaryotes, prokaryotes, and viruses. We will also explore the human genome in depth and learn how changes in the structure or number of chromosomes in the human genome can lead to certain genetic diseases. The image on this slide is a photograph of the model of the DNA molecule built by Drs. James Watson and Francis Crick in 1953. Drs. Watson and Crick used this model to depict their proposed structure for the DNA double helix. The hypothesized structure was derived from X-ray diffraction data produced by Drs. Maurice Wilkins and Rosalind Franklin. The model was constructed from metal scraps obtained from a machine shop. Drs. Watson and Crick published their proposed DNA structure in the journal Nature in 1953. For their work, Drs. Watson, Crick, and Wilkins were awarded the Nobel Prize in Physiology or Medicine in 1962. Each chromosome of a living cell is a DNA double helix. Embedded within the DNA sequence of each chromosome are the organism's genes. The chromosome can be thought of as the scaffolding within which the genes reside. An organism's set of chromosomes is called its genome. Genes are interspersed unevenly along the lengths of most eukaryotic chromosomes. Across the human genome, for example, there are gene-rich regions and gene-poor regions. The genomes of most eukaryotes are fairly large and often complex. In addition to the genes themselves, most eukaryotic genomes also contain a variety of non-coding, structural, and regulatory elements, including introns. Some of the genetic material also serves as a fossil record, a history book written in biological terms and handed down from generation to generation. In addition to the DNA inside the cell nucleus, eukaryotic cells have separate genetic material in certain organelles, such as mitochondria. Plants carry genetic material in organelles called chloroplasts. In general, organelles, prokaryotes, and viruses have greater biological constraints than the nuclei of eukaryotic cells on the tolerable sizes of their genomes. This is because of the small genome size that can be incorporated into the organelle, the bacterial cell, or the viral capsid. As such, the genes of mitochondria, bacteria, and viruses typically lack many of the complex non-coding elements commonly found in the genes of eukaryotes. The genes within an organism's genome are generally not evenly distributed along the length of that organism's DNA. The genes of mitochondria, bacteria, and viruses are often immediately adjacent to one another and frequently overlap. In eukaryotes, there are gene-rich and gene-poor regions within the genome, where non-coding spacer regions span the distance between gene-rich areas. Within the gene-rich areas, overlapping genes are common. Overlapping genes are different genes whose nucleotide sequences overlap along a segment of DNA. The nucleotide sequence of overlapping genes is read by RNA polymerases in 2 or more reading frames or from opposite strands of the DNA molecule. Overlapping genes enable encoding of different proteins within the same segment of DNA. Overlapping genes are found throughout nature, from bacteria to viruses to mammals, including humans. Viruses are not cells. They are genetic material encased in a protein coat called a virus capsid. Some complex viruses also surround themselves with a viral envelope or membrane made up of lipids and glycoproteins. The genetic and biological characteristics of viruses vary widely. Like bacterial genomes, the genomes of viruses are very small and typically do not contain introns. However, despite their small sizes, the genomes of viruses can be quite complex in that the genes of viruses often overlap or lie immediately adjacent to one another. Like bacteria, some viruses also encode multicistronic RNA molecules. Multicistronic RNA molecules are RNA molecules that contain the coding sequences of more than one protein, usually arranged in a sequential manner. A multicistronic RNA molecule encodes several proteins but is transcribed as a single RNA molecule. When the RNA is translated, one long polypeptide is made that is actually several proteins joined end to end. The polypeptide is processed by enzymatic cleavage to liberate the individual proteins from the larger polypeptide. In addition, many viruses demonstrate temporal regulation of transcription. For example, early and late genes are expressed at different times during the course of viral infection. There are two domains of prokaryotes, bacteria and archaea. Members of these groups do not have a cell nucleus or organelles bounded by membranes. Although there are exceptions, bacterial genomes do not typically have introns. On the other hand, the genomes of organisms in the domain archaea are sometimes a bit more complex and can have introns and other structural and regulatory elements similar to those found in eukaryotes. Prokaryotic genome sizes can vary widely. Some may be as small as a few 100,000 base pairs, some may be as large as several million base pairs. Like the extensive diversity in life forms found within the eukaryote domain, the genomes of eukaryotes also vary greatly in size and composition. For example, the yeast Saccharomyces cerevisiae contains a genome 12.5 million base pairs in size, while the fern Psilotum nudum contains a genome 250 billion base pairs in size. Further, most eukaryotic genomes encode complex genetic elements that preserve and maintain the structure of the genome, regulate the transcription of genes of eukaryotes, and also serve as a fossil record for the species. In addition, the genetic material of eukaryotes is organized into one or more linear structures called chromosomes. Mitochondria are organelles within eukaryotic cells. Mitochondria are the power plants of cells. They provide cells with the chemical energy needed to perform the metabolic tasks associated with life. It is widely believed that mitochondria are the remnants of ancient energy-producing symbiotic bacterial cells that were assimilated into eukaryotic cells. Similarly, chloroplasts in green plants are believed to be remnants of photosynthetic bacteria. The mitochondrial chromosome encodes 13 proteins and 2 ribosomal RNAs, and also 22 transfer RNAs. In addition, mitochondria obtain many of their proteins from genes encoded in the nuclear genomes of cells. Nuclear encoded proteins are imported into the mitochondria after translation. The haploid human genome consists of 23 different chromosomes. The diploid human genome consists of 23 pairs of chromosomes. One of each pair of chromosomes is inherited from each parent. For example, each of us carries 2 copies of chromosome 1, one from our mother, the other from our father. The first 22 pairs of human chromosomes are called autosomes. The 23rd pair of human chromosomes determine gender. Human females carry 2 X chromosomes, one inherited from the mother, one inherited from the father. Human males carry 1 X chromosome and 1 Y chromosome. Males inherit their X chromosome from their mothers and their Y chromosome from their fathers. In addition to the nucleus, the mitochondria of human cells also carry 1 or more copies of a small circular DNA molecule. The mitochondrial chromosomes are inherited through the egg. Sperm do not contribute mitochondria to embryos. As a result, genetic diseases caused by mutations in genes on the mitochondrial chromosome are passed through the maternal lineage, that is, from mothers to all their children. Children of fathers affected by mitochondrially transmitted genetic diseases are not at risk for those disorders. However, because mitochondria also import proteins encoded in the nucleus, the inheritance of diseases affecting mitochondria do not always follow a maternal pattern of inheritance. In general, the observable effects of mitochondrial mutations are believed to be a reflection of how sensitive particular tissues are to energy metabolism. The haploid human genome, in its 23 chromosomes, contains approximately 3 billion base pairs of DNA. The diploid human genome, with 23 pairs of chromosomes, contains about 6 billion base pairs of DNA. Only about 1.5 to 3% of the human genome codes for proteins. Much of the rest of the human genome serves regulatory and structural functions. The exact number of genes carried by humans is not yet known, but it is likely to be somewhere between 35,000 and 100,000 genes. Humans are more than 99% identical to each other at the DNA level. Only identical twins possess identical genomic DNA. However, it is the 1% variability in our DNA that makes each of us unique. In recent years, the variability in human DNA has been used to study human history and trace the migrations of humans out of Africa as we spread across and populated the Earth. What we have learned from our DNA is clear. Every human on the planet today is related to every other human. We are all descended from the same trunk of the human family tree, brothers and sisters separated only by time and geography. The photograph in this slide is of a stack of Houston telephone books spiraled to mimic the shape of the DNA double helix. The stack of telephone books contains approximately the same number of characters as the haploid human genome contains base pairs. The stack illustrates the amount of information contained within the human genome. If the DNA contained within a single human cell were laid out lengthwise, with each chromosome arranged end to end, the DNA would be 5.8 feet or almost 2 meters long. However, it would be very thin. At only 24 angstroms wide, it would take 26 million DNA molecules arranged side by side to measure 1 inch wide. To fit into the microscopic nucleus of a human cell, the length of DNA must be packaged very precisely. In the first level of packaging, the DNA double helix is wound tightly around proteins called histones. Histones are made up of 8 component proteins and are often referred to as histone octamers. The packaging of DNA around histones gives DNA a beads-on-a-string appearance. Histone-packaged DNA results in a 10 nanometer fiber. Remember, the native DNA is only 24 angstroms wide, so this 100 angstrom or 10 nanometer fiber is 4 times wider and quite a bit shorter than the native DNA double helix. The histone-packaged DNA is further wound into 30 nanometer fibers. These fibers are then packaged into loops of DNA thought to be anchored to the cytoskeleton within the nucleus of the cell. DNA that is completely packaged is referred to as chromatin. The expression of genes within the human genome is controlled by a number of different mechanisms. Promoters, enhancers, and silencers are segments of DNA that regulate the rate and amount of transcription or expression of nearby genes. Promoters are generally located immediately upstream from the first exon of a gene. Enhancers are genetic elements that increase transcription of associated genes. Silencers are genetic elements that suppress transcription of associated genes. Enhancers and silencers can be located some distance away from a promoter. They can be upstream or downstream from the promoter, and they can be effective even at considerable genetic distances. Methylation is another mechanism of gene regulation. In methylation, portions of the DNA molecule, particularly regions of the DNA that can control transcription, are chemically modified through the action of enzymes called DNA methyltransferases. DNA methyltransferases attach a methyl group to the 5th position carbon of cytosine bases, typically when cytosines occur in the context of CG dinucleotides. These CG dinucleotides are sometimes referred to as CPG islands. Methylation of DNA usually results in the silencing or suppression of gene expression. Alternatively, a lack of methylation is usually associated with an active state of gene expression. Methylation is crucial for the regulation of gene expression and control of cellular growth throughout life, from embryonic development to adulthood. Methylation is also crucial for X chromosome inactivation and imprinting. Imprinting is a form of gene silencing whereby individual genes or groups of genes are methylated during gametogenesis to impart parent of origin effects on gene expression. For example, if a gene is maternally imprinted, that is silenced in the maternal copy during female gametogenesis, only the paternal copy of the gene is expressed in the child. Paternal imprinting of genes also occurs. During gametogenesis, parental imprints are erased and reset in a manner consistent with whether the gametogenesis is occurring in a male or a female. A number of human genetic diseases occur because mutations in imprinted genes occur, or mutations occur in the genetic control sequences responsible for setting, resetting, or controlling imprinted regions. Such diseases include Angelman syndrome and Prader-Willi syndrome. These diseases occur because of mutations in imprinted genes or errors in imprinting of genes on chromosome 15. These disorders can also be caused when errors in mitosis or meiosis produce a child who inherits both copies of his or her chromosome 15 from a single parent. This circumstance results in inheritance of only imprinted copies of the genes. This phenomenon is called uniparental disomy, or UPD. Another type of genetic regulation is exerted at the level of RNA processing through a mechanism called alternative splicing. In alternative splicing, certain exons of a gene may be alternately included in the final messenger RNA product. In this way, inclusion of particular exons only in certain tissues or only at certain times during development permits cells to modify the performance characteristics of proteins in particular cells or tissues. As shown in the graphic, this precursor RNA contains 4 exons. It is alternatively spliced into 3 different isoforms. Isoform 1 contains all 4 exons. Isoform 2 contains only exons 1, 2, and 4. Isoform 3 contains only exons 1 and 4. Various factors, including DNA sequences, also contribute to the rate of production and stability of messenger RNAs. By controlling the rate at which new messenger RNAs are transcribed from a particular gene, and the decay kinetics of a messenger RNA, that is, the rate at which a messenger RNA is degraded within a cell, cells can exert control over the amount of protein that is made from a messenger RNA. This influences the level of the particular proteins within cells and tissues. Different messenger RNAs have different decay kinetics, meaning that some are degraded very quickly and others are degraded much more slowly. For example, the messenger RNAs of many cellular growth factors and transcription factors are degraded very quickly, resulting in tight control of the temporal aspects of their cellular signaling properties. On the other hand, messenger RNAs that encode enzymes can be much longer lived. During X inactivation, most genes on one of the X chromosomes of a female are inactivated in a mechanism that provides dosage compensation to equalize the level of gene expression of most X-linked genes in females to that of males who have only one X chromosome. This process, often called lyonization, results in the formation of a bar body, a highly compacted, inactive X chromosome. In the photograph, the bar bodies are shown by arrows. The form of DNA in the inactive X chromosome is called heterochromatin. In human females, one X chromosome or the other is inactivated in each cell early in development. As a result, some human female cells have one X chromosome active, while the rest of the cells have the other X chromosome active. Typically, X inactivation is random or near random, meaning that roughly half of a female's cells express the genes of one X chromosome, and the other half of her cells express genes from the other X chromosome. However, in some females, X inactivation is not random, but rather is skewed. In cases of non-random or skewed X inactivation, all or most of the cells of a female express X-linked genes from only a single X chromosome. In reality, non-random X inactivation may be less representative of the process of X inactivation than it is of reduced survival of cells with a particular X chromosome inactivated. In these circumstances, it makes it appear that X inactivation was non-random. In some cases, females who carry X-linked genetic diseases can be affected by those diseases if X inactivation is not random. For example, if a structural abnormality disrupts the pattern of X inactivation, most or all of a female's cells will express genes from a single X chromosome. If there are genes on that chromosome that are disrupted by the structural abnormality, and those genes cause a disorder, the female can show symptoms of the disorder. Further, for many X-linked disorders, females will show mild to moderate symptoms. This milder manifestation of an X-linked disorder in females may be related in part to the fact that most females have, in addition to a large number of cells expressing the abnormal gene, a large number of cells expressing the normal gene. Symptoms remain milder than those seen in affected males who carry no normal copy of the gene because they only carry a single X chromosome. Some genes on the inactive X chromosome escape inactivation and remain active in females. These genes are believed to be responsible for many traits of normal human females. The lack of these genes in females missing all or a portion of their second X chromosome as occurs in many cases of Turner syndrome, are thought to be associated with many of the clinical features of the syndrome. Karyotyping is a laboratory technique that allows microscopic visualization of the number and gross structure of the chromosomes within a cell. The image in the slide shows the karyotype from a normal human male. Note 1X and 1Y chromosome. This karyotype would be abbreviated 46, XY. 46 for the number of chromosomes, XY for the sex chromosome makeup. If this karyotype were from a normal female, there would be 2 X chromosomes and no Y chromosome. A normal female karyotype would be designated 46XX. The signs and symptoms of genetic disorders caused by disruptions in the structure or number of chromosomes in the human genome are extremely variable and typically are related to the particular chromosomal abnormality involved. In the case of structural rearrangements of chromosomes, most effects are likely due to disrupted expression or regulation of genes at or near the sites of structural rearrangements. In the case of chromosomal aneuploidies, the condition of having more or less than the normal number of chromosomes, alteration of the normal copy number of the genes on the involved chromosome are likely the cause of most of the abnormalities. The karyotype in this slide is that of a female. Note the 2 X chromosomes and the lack of a Y chromosome. Note also that this individual carries 3 copies of chromosome 21. This chromosomal aneuploidy is called trisomy 21 or Down syndrome. Down syndrome is caused by the presence of an extra copy of chromosome 21. This karyotype would be abbreviated 47, XX, plus 21. Down syndrome is characterized by mental retardation, speckling of the irises of the eyes, poor muscle tone, also known as hypotonia, a characteristic appearance of the face, cardiac abnormalities, loose skin in the back of the neck, and fine, soft hair. Individuals with Down syndrome are also prone to leukemia, premature aging, and Alzheimer's disease. Down syndrome affects approximately 1 in every 800 live births. About 75% of Down syndrome fetuses spontaneously abort during gestation. Of babies born with Down syndrome, most carry an extra copy of chromosome 21 that is a freestanding chromosome. The extra chromosome occurs as a result of an error in cell division during meiosis. Meiosis is the process by which germ cell precursors divide to create eggs and sperm. During meiosis, there are occasional sorting errors, wherein the chromosomes do not get properly sorted to dividing cells. This can result in a sperm or egg cell with an improper number of chromosomes. Most of the meiotic non-disjunction events that result in Down syndrome occur during maternal meiosis. In some cases of Down syndrome, however, the extra copy of chromosome 21 is not freestanding, but rather attached to another chromosome. Chromosomal rearrangements in which all or a portion of a chromosome become attached to another chromosome are called translocations. Translocation cases account for approximately 3% of Down syndrome cases. The source of the extra chromosomal material, whether it is derived from a parental translocation or as the result of a meiotic error, is important for estimating the chance that another child with Down syndrome will be born in a family. The risk for a woman to have a child with Down syndrome rises with age and increases significantly after age 35. This phenomenon is called a maternal age effect. Despite this maternal age effect, however, most babies with Down syndrome are born to women under 35. This is because more babies are born to women under 35 overall than are born to women over 35. The image in this slide is a karyotype of an individual that is a female. Note the 2 X chromosomes and the lack of a Y chromosome. This individual carries 3 copies of chromosome 13. This is a chromosomal aneuploidy called trisomy 13. This syndrome is caused by the presence of an extra copy of the genes on chromosome 13. This karyotype would be abbreviated 47XX plus 13. Babies with trisomy 13 can have a variety of congenital abnormalities, including incomplete development of the forebrain, olfactory, and optic nerves, seizures, apnea, mental retardation, deafness, microcephaly, which is a small head, sloping forehead, cleft lip and/or cleft palate, skin abnormalities, congenital heart defects, additional fingers and toes, prominence of the heels of the feet, thin or missing ribs, and a number of other abnormalities. Trisomy 13 occurs in approximately 1 of every 5,000 live births. More than 95% of fetuses that are trisomic for chromosome 13 spontaneously abort during gestation. Most babies with trisomy 13 that are born alive die within the first few months of life. About 20% of cases of trisomy 13 are caused by translocations, when all or a portion of chromosome 13 is attached to another chromosome. More often, the extra copy of chromosome 13 is the result of an error in cell division during meiosis. Most of the meiotic non-disjunction events that result in trisomy 13 syndrome occur during maternal meiosis. The source of the extra chromosomal material, whether it is derived from a parental translocation chromosome or as the result of a meiotic error, is important for estimating the chance another child with trisomy 13 syndrome will be born in a family. In this slide set, we have discovered that genomes contain the genetic information of organisms. Large genomes undergo extensive packaging to facilitate their enclosure within cells. The expression of genes within an organism's genome is tightly regulated by a variety of mechanisms. These mechanisms ensure proper growth and development of the organism. Alterations in an organism's genome can lead to disabilities. A number of human genetic disorders are caused by changes in the structure or number of human chromosomes.